Good morning, students. We are going to start our 5th lesson on cell biology. Students, if you remember, in our last period, I have given you some questions to work as homework. In this period, we are going to answer these questions as summary, and we will start our lesson. The first question says, what is the difference between simple diffusion and facilitated diffusion? You can answer this question as you understand how you can differentiate the two types of passive processes, but you can answer the question as follows. Simple diffusion not use transport protein, but facilitated diffusion use transport protein. Both are passive processes, but simple diffusion use transport proteins, and facilitated diffusion do not use transport. It uses transport protein. The second question, what is the difference between passive process and active process? Students, you can list at least two differences between these two processes. Passive process, the movement of substance is from high concentration to low. In active process, the movement is from low to high. Secondly, passive process do not use cell energy, whereas active process use cellular energy to transport particles against concentration gradient. If you answer like this, you will get full marks. Okay. In our previous class, if you remember, we start discussion on passive process, and we have seen that passive process is type of transport in which particles transport from high concentration to low, and in the absence of cellular energy, the process will proceed. And this process can be of three types. One of the process was simple diffusion, where we have discussed in our previous lesson, and the other one is facilitated diffusion. To remind you what we have seen in our last period, simple diffusion transport particles from high to low concentration, and this process can be affected by three factors. Can you remember these factors? Okay, these factors are concentration gradient, surface area, and thickness of membrane. Again, in case of facilitated diffusion, particles, as like that of simple diffusion, transport from high concentration to low concentration. The difference is, in facilitated diffusion, the cell use transport protein. Why? Because the particles are large in size, they cannot cross the plasma membrane. And this process is also affected by three factors. One of the factors was the number of channel protein and carrier protein, or in all transport proteins. Why? If the plasma membrane has large surface area with low number of carrier and channel proteins, the process will be affected. The second factor is concentration gradient, and the other one is thickness of membrane. The third passive process is osmosis. Osmosis is movement of water molecule. Bear in mind that this process is movement of water molecule from region of high concentration to region of low concentration through partially permeable membrane. Again, if you remember, on simple diffusion, the membrane may be partially selectively permeable, or it can be permeable membrane. To occur simple diffusion, it can occur in both type of membrane. But in case of osmosis, the first point, the particles are water molecules. The second point, there must be selectively permeable membrane. So what will happen in the process? During osmosis, the water particles will cross the plasma membrane from region of high water concentration to low. In this part, when we talk about high water concentration, you have to remember that we are referring to the water potential of the system. Water potential is, students, water potential is the concentration of free water molecules. Again, you have to bear in mind this free water molecules in the system. Therefore, osmosis occurs from high water potential to low through partially permeable membrane. As this diagram shows, it is an example that helps us to understand what osmosis is. The smaller circles or the blue circles represent water, and the larger circle represents sugar. And water molecule cross the plasma membrane from high concentration. As you see here, they are found high, and they will cross to the next part through the partially permeable membrane, and this process is known as osmosis. In osmosis, as we said, the water potential is important for the process. Why? Because when particles are dissolved in solution, the amount or the number of free water molecules in that solution decreases. From your chemistry, you know that when solutes dissolve by solvent, for example by water, since it is universal solvent, the water molecules will bind to the particles of solutes like this in order to dissolve the solute, and when they bind to the solute particle, they will form what we call hydration layer. When they form this hydration layer, the number of free water molecules in the system decreases. Due to this, when biologists measure water potential of different systems, pure water is found to have the highest measurement of water potential, that is 0. Pure water has the highest water potential, that is 0. This is because in pure water there is no mixed solute particle in the system. For that reason, all the system, or 100% the system, contain free water molecules, and it is 0 Pascal. Water potential is measured by Pascal. It is unit used to measure pressure. But compared with other solutions or system, the water potential of liquid water is the highest. This is because if take intracellular solutions, or if take body fluid, or any solution, it must have measurement of water potential less than 0, which means the water potential of any system becomes negative. Since 0 is the largest number, then the rest become negative. So all solutions have negative water potential. That is because the water molecules bind with solute particle and decrease the number of free water molecules in the system. Students, would you please explain why pure water has the highest water potential? Let me give you 20 seconds to think and write your answer. Okay, welcome back, students. When we explain why pure water has the highest water potential, we can say that pure water has the highest water potential because it is not mixed with any solutes. If you answer like this, you got the correct answer. The answer will be pure water has the highest water potential because it is not mixed with solutes. That's why we consider the water potential of pure water as the highest potential. Therefore, by now we have the ground to define osmosis in terms of water potential. So when we define osmosis in terms of water potential, it is defined as the movement of water molecules from region of high water potential to region of low water potential through partially permeable membrane. You have to put these important terms in your definition. Osmosis is a process by which water molecules can transport from region of high water potential to region of low water potential through partially permeable membrane. This is how we can define osmosis. Okay, students, in our previous class, when we talk about simple diffusion and facilitated diffusion, we have said that these two processes, facilitated diffusion and simple diffusion, are also affected by factors, different factors. And again, osmosis is also the same with those types of transporters. And this process is also affected by the same type of factor with a slight difference. The factors that affect osmosis are surface area to surface area of the membrane is one of the factors that affect rate of osmosis, and the difference in water potential between the two areas outside the cell or inside the cell. The difference can also affect rate of osmosis. But in our previous class, we have said that the concentration difference between the two areas. But in our osmosis, it is the water molecule that transport through the plasma membrane. For that reason, rather than saying water concentration, it is preferable to use the water potential of the system. And the other one is the distance the molecule travels. The water molecule travels can affect the process of osmosis. Okay, up to now we have seen how osmosis occurs and how we can understand the effect of osmosis. But biologically, to understand the effect of osmosis, we have to use some comparisons. When we compare or depending on the water potential of the cell with its environment, the cell with its environment, the movement of water can be determined by the difference in water potential between the two solutions, the cell sub-solution, I mean the cell and its environment. Based on this, we have three types of solution. The first solution is isotonic solution. Iso means the same and having the same water potential as the cell, which means if we say isotonic solution, the solution has the same water potential with that of the cell. And hypertonic solution, in hypertonic solution, the solution have lower water potential than the cell. The lower water potential than the cell, or it has negative water potential than the cell. And hypotonic solution having higher water potential than the cell. But the difference is in hypotonic solution, imagine we put a particular cell in a solution, and the solution can be hypertonic or hypotonic. If the solution is hypertonic, what will happen? That hypertonic solution has negative water potential compared with the cell water potential. And if we put hypotonic solution, since all solutions have negative water potential, if we put cells under hypotonic solution, that solution has less negative water potential compared with the cell water potential. But in this case, again, the water potential difference between the cell and the solution can also determine the movement of water. For have more understanding, let's have these two diagrams to understand the process of osmosis in hypertonic solution and in hypotonic solution. In the first case, the solution is hypertonic. When we say hypertonic solution, it is to mean that the cell has high water potential and the solution has low water potential. If you look at the cell, it has 4% sugar and 96% water. If you look at the solution, it has 7% salt and 93% water. This indicates when we compare the water potential of the cell with that of the water potential of the solution, the cell has more water potential than the solution. So what will happen? In our osmosis, in our definition of osmosis, we all know that water moves from high water potential to low water potential. Osmosis occurs from high water potential to low water potential. That means from the cell to the solution. Or the cell will lose water to the solution in case of hypertonic condition. And on the second case, in hypotonic solution, hypo means higher, and in this solution, what will happen? The cell has low water potential, but the solution has high water potential. In this condition, water moves from the solution into the cell. Why? We know that again, in definition of osmosis, water moves from high water potential to low water potential, that is from the solution to the cell. This symbol is psi, which is represent water potential, and in this case the cell will gain water. The cell will gain water from different direction, and it become a swell, or it will gain water and it becomes swell. But in case of hypertonic solution, water moves out of the cell in all direction, because the cell has high water potential compared with the solution. This is how you can understand what will happen in hypotonic and in hypertonic solution. For further understanding, again, we can use different types of cells in hypertonic and in hypotonic solution to understand the process. In hypertonic solution, if you put animal cells in hypertonic solution, what will happen? The cell lose water. The cell will lose water by osmosis, and it becomes shrinks. And if we put animal cells in hypotonic solution, it will gain water by osmosis and it will swell. But since animal cells have no cell wall, swelling in animal cell will lead to rupturing of the plasma membrane. And we call this process bursting. The cell will burst. If bursting occurs in human red blood cell, or what we can say, if you put human red blood cell under hypotonic solution, the cell will gain more water, and since the human red blood cell is animal cell, it has no cell wall that can give support or that can give strength, the cell will rupture or the cell will burst, and this condition is known as hemolysis. It is known as hemolysis. It is unique for human red blood cell. Again, in case of plant cell, if you put plant cells under hypertonic solution, like that of animal cell, plant cells also lose water and they will shrink, but shrinkage in case of plant cell can be of different type. If the cell lose water simply due to loss of water and shrinks, we call it flaccid. But excessive loss leads to a process known as plasmolysis. The difference is, if the cell is plasmolysed, the cell membrane and the plasma membrane will separate and concentrate at one part of the cell. You can take example, wilting in plant. In case of flaccid cells, they become weak and fail to support their leaves and so on. In hypotonic solution, plant cells gain water and swell, but what makes a difference with that of animal cell is plant cells do not burst, but they become turgid, and we call the condition turgidity. This is very important for plants, especially woodless plants or those plants with no woods. This give support and strength for the plant. So up to now we have seen how osmosis occurs and the effect of osmosis on plant and animals. So you have to remember the effect of osmosis in plant cell and the effect of osmosis in animal cells. Okay, now I have questions that can be answered based on osmosis and simple diffusion or passive process. List some examples of molecules that are transported by simple diffusion or facilitated diffusion. This is the lesson in our previous class. Simple diffusion transport what type of substances? It transports small, nonpolar and lipid soluble substances. And in case of facilitated diffusion, substances transport like polar molecules, large molecules and charged molecules. If you answer like this, you are correct. The second question says, if 0.5% salt solution is assumed to be isotonic to a cell, in which of the following solutions do this cell lose water? You have given 4 choices and you are expected to choose one of the 4 choices in which the cell lose water. Can you try this in 30 seconds? Okay, let's return back to our question. If 0.5% salt solution is isotonic for the cell in order to lose water, it must be under what type of solution? Under hypertonic solution. A solution hypertonic for 0.5% salt solution must have more salt content than this solution. Why? Because we have said that 0.5% salt solution is isotonic for this particular cell. Therefore, when we come to the choice A, 0.05% salt, this salt solution cannot be hypertonic for our ideal cell, because having 0.05% salt means more dilute solution. If you look at 0.7% salt solution, it contains more salt than the ideal isotonic solution. So this can be considered as the answer. If you look the other choice, 0.01%, it is more diluted, or it has more water potential than the cell solution, so it cannot be considered as hypertonic, or it cannot make the cell to lose water. Therefore, our answer will be B. If 0.5% is isotonic to the cell, any solution that has greater solute concentration or less solvent concentration become hypertonic to this cell. So the cell will lose water under the choice B. Is that clear, students? Okay, let's move on to the next question. If the above cell, it says, if the above cell is plant cell, it becomes what? In our previous question, our cell was placed under 0.5% salt solution, and if this cell lose water under 0.7% salt solution, that means the cell is under hypertonic condition. So if this cell is plant cell, what will happen to the cell? You have choice, so you can answer this by 50 seconds. I will expect the answer within 50 seconds. Okay, I hope you have done this question correctly. If you answer, the cell becomes plasmolysed, that will be correct answer. Let's move on the third question. If the above cell, again, is animal cell, it becomes burst in a solution with what? You have given 4 choices, so you can answer your question. Look at the choice carefully and try to answer the questions. Okay, the answer becomes pure water, because if the cell is animal cell and it burst or it gain more water if it is placed under pure water. The other solutions cannot lead to bursting. Okay, by this we have finished what passive process is. Now let's see the second type of process, that is active process. Active process has 3 types, and we will see these 3 types of processes in this period. The first one is active transport. Active transport, or it can move particles against concentration gradient or from low concentration to high concentration by using energy in the form of ATP and transport proteins. These transport proteins are found in the cell plasma membrane. And in active process, in active transport, always molecules transport down concentration gradient. When we say down concentration gradient, it is to indicate that the particles move from low concentration to high concentration. As you see here on the diagram, particles move from extracellular fluid to intracellular fluid by using energy. This energy is used by the cell to transport the particles into the cytoplasm. The second type of active process is endocytosis. As its name indicates, particles move into the cell. Here we have example of endocytosis. As you see here, the particles move into the cell through the plasma membrane, and the plasma membrane will form a sort of vesicle to transport the particles. And this endocytosis can be of three types: phagocytosis, which means cell eating; pinocytosis, that is cell drinking; and the third one is receptor mediated endocytosis. Phagocytosis takes large particles and solid particles into the cell, whereas pinocytosis take in smaller particles compared with the particles in phagocytosis. But in receptor-mediated endocytosis, the particles to be entered into the cell must be specific, because the receptors are specific for a specific type of particles. As you see here on the membrane, there are receptors which can only accept particular proteins or particles to move into the cell, And the last active process is exocytosis. As its name indicates, exo means outside the cell, and in this process, particles move out of the cell by secretory vesicles. And the cell uses this process to release hormones, enzymes, for example, sweets, and other waste products. Here we have the diagram that shows exocytosis. The particles will collect in form by vesicle and they become removed from the cell. This is how exocytosis occurs, and we have seen a lot on transport in cell as passive process and active process. You have to read your textbook in more detail to understand, and this brings us to the end of the lesson. Thank you for listening.